The medical laboratory technologist is an important member of the healthcare team because without their skills or interpretation of the patient's results, the physician would be without important information to make an accurate diagnosis. There have been many breakthroughs and discoveries within this field that required meticulous research.
In 1901, Karl Landsteiner discovered ABO blood typing. This was a critical step for saving many lives in the transfusion process. Research in this field of study is currently being conducted to further expand the knowledge of this important piece of medical laboratory technology. Examples include the introduction of official allele nomenclature and a potential breakthrough of creating ABO universal transfusion blood. Knowledge of ABO typing has been the basis of research for many aspects of the lab and will continue to save millions of lives as research and knowledge in this field expands.
Kary Mullis first published and patented the polymerase chain reaction test in 1985. This test amplifies the amount of DNA and RNA found in humans or microorganisms. His 2 years of research informed him of the different conditions needed to run the test by altering the temperature, sensitivity of radioactive gel, the amount of enzyme, and the amount of cycles needed. This research has benefited medical laboratory science because the amount of time taken to diagnose a patient's illness is decreased. This test also has implications in the diagnosis of genetic diseases.
Standard precautions were developed and implemented in the late 1990s in rebuttal to the increase of acquired HIV infections in healthcare professionals. Standard precautions are defined as a set of implemented guidelines to reduce the incidence of healthcare-associated infections. They include the use of personal protective equipment, safe disposal of sharps, and an understanding of standard principles of healthcare-associated infections. In other words, MLTs now treat all samples as potentially infectious. Standard precautions were developed through multiple reviews of experimental and non-experimental research within hospitals. Since the implication of standard precautions in North American hospitals, many MLTs have avoided the contraction of potentially infectious diseases.
In recent years, medical researchers have recognized the significance of genetic components when dealing with the world's most serious diseases. Since the mapping of the first disease gene, there have been many more discoveries of specific cell markers for certain diseases, and we now have genetic screening for more accurate patient diagnosis. It will take many more years of research before we can fully understand the human genome, but research into medical genetics has already changed the dynamics of the laboratory and will continue to do so as new technologies are introduced to analyze cell markers. As the majority of clinical decisions are based on data from laboratory testing, research is a crucial part of the medical laboratory for the continuation of optimal patient care.